Hello and welcome. Welcome to this wonderful virtual symposium to launch the Barts Centre for Squamous Cancer. You're most welcome. I'm Paul Coulthard, Dean of the Institute of Dentistry here at Barts Centre, London School of Medicine and Dentistry at Queen Mary University of London. We have a number of speakers for you this afternoon, some from our own university and some from elsewhere in the UK and even further afield. So I'm delighted that you'll be hearing a little later from our keynote address speaker, Professor Jennifer Grandis, Professor of Head and Neck Cancer Surgical Research at the University of California, San Francisco. Thank you for making the time, Jennifer. We really appreciate it and look forward to your address. Thank you. I am Professor of Oral and Maxillofacial Surgery and I'm an oral surgeon. So I know more about oral cancer than I do other squamous cancers, such as those of the esophagus, skin, lung, and cervix. But you'll be hearing more about what's common across these cancers a little later. And a big, unique aim of our centre is to increase research into common pathways of genomic, transcriptomic, and clinical significance across these squamous cancers. You can see here the state of play regarding oral cancer. And you can see that the numbers are sadly increasing and that, unfortunately, awareness of this disease is not as common as it ought to be. We know that there's increased oral cancer numbers in areas of deprivation, and particularly so for men. We know something of the risk factors. We know that smoking, alcohol, diet, viruses, and palm are involved, but we don't understand enough, and prevention, detection, and treatment are poorly effective. You can see here the WHO figures from last year about oral cancer and its significance around the world. And in particular, the mortality picture on the right shows the high prevalence and unfortunate death rates in Asia. Our dental school is situated in the East End of London. It's a great dental school. We've risen through the ranks of late, so we're currently 11th in the world. And I have to say, I'm very proud that in our citations, we are first in the world. We also did very well in the UK government's research assessment framework last time round, and we hope to do well again. Our research is structured in these three ways, and you can see that we do already have oral cancer amongst these. We have a number of good researchers undertaking wonderful research, but our aim is to increase collaboration across our faculty, university, and beyond. Our mission for oral cancer research within our Institute of Dentistry is to answer the overarching research questions as to who, how, and why patients develop oral cancer in the East End of London by a comprehensive database, a biorepository, genomic and transcriptomic analysis, and clinical trials, and to, of course, extrapolate that for the global good. We have around our hospital and school a large Bangladeshi population with almost half the local population. Whilst born in England, we know that some of these risk factors will abound, and sadly and inevitably, there's a high incidence of oral cancer. When I moved to London 2.5 years ago, this was one of the key reasons why I chose to do so. And I, like many who work here, that East End London population gives us a particular opportunity to do something really worthwhile. The opportunity to do something through our research to improve the health of those locally, but also globally. A little bit more about oral cancer. You can see that the treatment is largely surgical, sometimes followed by radiotherapy. Oropharyngeal cancers tend to present later because of their location and more likely to be at an advanced stage, in which case they may be managed with more radiotherapy and chemoradiation. But generally, surgery is the mainstay. Of particular note, as you can see here, is the survival rate, which is unfortunately very poor and has not sadly increased over the decades. So we need to do something about it. Surgery for oral cancer has its own problems. There are huge, significant functional consequences around basic requirements of human beings to have fulfilled and happy lives. Eating, drinking, speaking, hearing, facial expression can all be affected. Depends, of course, on the nature of the surgery and also the extent of it, and of course, based on when the patient presented and how significant the disease was. But effects on appearance and identity alone can be really unfortunate consequences for these patients. So we need to increase the survival and the quality of life for these patients. Now, I don't mind sharing with you that just over a year ago, I suffered cancer of the prostate, underwent surgery very effectively, and I'm doing really very well. And I have to say, I was less daunted by that, probably because there's been more research in that area and the surgical sciences have advanced. So this is an interesting slide that shows that there's been a considerable mismatch between global funding and disease burden. Some cancers are funded at a higher level than their relative burden, such as prostate cancer and breast cancer, and some cancers are underfunded, for example, oral cancer and esophageal cancer. So this is where Barts Charity comes in, and we're delighted that they've generously awarded just over £2.6 million to create the Barts Centre for Squamous Cancer. So this will be dedicated to improving detection, treatment, and improving the quality of life for patients with squamous cancers through cross-institute calibration and bringing together research groups with diverse expertise from across our School of Medicine and Dentistry to tackle the problems. It will be dedicated to work with the population of East London and to tackle important questions for them. And as I said earlier, to extrapolate that to the great benefit of those suffering these diseases globally. So these are my colleagues I'd like to introduce you to who have been critical to us obtaining this funding. Irene Lee, Professor of Cellular Molecular Medicine, who has done most of the work on our application to Barts Charity. Catherine Harwood, Professor of Dermatology, and David Kelsall, Professor of Human Molecular Genetics. And more, of course. So you can see here in our structure that we have many other individuals who have been key to putting together our plans to tackle these squamous cancer diseases. And some of them you'll be hearing from in this symposium. So I'm also pleased to say that we're working in collaboration with the Oral Cancer Trust, which is a charity. And of course, we want to work significantly with patients, those who have had this disease, have the disease, or know people who have the disease. So our next steps are to increase our research capacity by recruitment of new additional top researchers. We already have a number of researchers across our faculty, as I've already described, but we wish to recruit 3 outstanding new researchers in the areas of molecular carcinogenesis, translational cancer research, and the epidemiology of oral cancer. We're also setting up 3 core facilities with staff. One of them is a tissue bank with clinical database, also a laboratory support for in vitro and in vivo models, and bioinformatics core support, so that we're well set to proceed. So thank you so much for listening to my introduction.
So it is such a pleasure to be here. I only wish I was there in person, not only because of the time difference, but your building looks so cool. And during the pandemic, I must confess, I've become an absolute addict for British television shows. So I would just love to be in England and tour London and see all my favorite spots and characters. But it's really a pleasure to be here today. I want to start by emphasizing, from my point of view, the incredible importance of team science. We don't do any of these things by ourselves. Academic medicine often emphasizes the glory of the individual in terms of career advancement, but it's my great pleasure now to co-direct a research group with my basic science partner, Dan Johnson, and my head and neck surgical partner, Patrick Ha, and together we are able to host a nice diverse group of trainees, and this is February in San Francisco, so that's the best part in some ways about living there. It's always lovely weather. I need to disclose that I have an investigator-initiated trial with support from Insight Pharmaceuticals. I will not talk about that today. I do some scientific consulting for startups in the Bay Area, and I will speak a little bit about a drug that I've co-invented that targets the transcription factor STAT3. So I have some patents on that. So I don't need to belabor this point for this audience. Your center sounds fantastic. Jill Siegfried and I, who is a lung cancer biologist, have always maintained that squamous cell cancer of the lung and of the oral cavity are very similar disease. And I'm sure squamous cells everywhere are very similar disease. We get balkanized by the types of surgery we do in the patients that we see. Today, I'm going to focus my talk mostly on oral cavity and then oropharynx and hypopharynx and larynx cancers, squamous cell carcinomas of the head and neck. But one thing that's really important to remind all of us about, there are very few virally associated cancers. And nasopharyngeal cancer caused by Epstein-Barr virus is endemic in southern China. And moving to San Francisco from Pennsylvania, I realized that the really large Cantonese population made nasopharyngeal carcinoma caused by EBV relatively common. And I will tell you a story that I'm sure you're all familiar with about the emergence of human papillomavirus as an important cause of oropharyngeal cancer. And those of you who are clinicians realize when you look in the back of someone's nose or mouth, there's no line separating the nasopharynx from the oropharynx. And so I think it's really compelling biologically that we have these two juxtaposed anatomic regions that are ripe soil for viral-induced carcinogenesis, something we really need to learn about. There are two types of squamous cell cancers that arise in the head and neck. The most common, the type that I saw a lot of when I was training, is associated with smoking. Now, I do love this picture, but I will point out that if you get to be 100 years old and you're still smoking, you're probably not going to die of an oral cancer. But the HPV-associated cancers of the head and neck have really emerged as an epidemic. And to underscore that point, you know, this is an older slide, but this line just keeps going up and up and up. The number of HPV-positive throat cancers in men passed the number of cervical cancers in women in the US a few years ago. And last year, according to the National Cancer Institute SEER data, throat cancers in men associated with HPV was the #1 fastest increasing cancer in the United States. And so when we look at our specimens, probably 80 to 85% of oropharyngeal cancers are associated with human papillomavirus. I will just say one final thing about this, then delve into the biology. This is, from my point of view, an entirely preventable cancer. I hope that in the United Kingdom and in Europe, you do a much better job of vaccinating your boys and girls before they're exposed to HPV. It's quite chaotic in the United States, and the vaccination rates overall for girls are about 2/3, but for boys, it's still only about 1/3 of our boys are being vaccinated against HPV prior to exposure, which from my point of view as a cancer doctor makes absolutely no sense. We have a safe and effective vaccine that can prevent a lethal and morbid cancer. It's, from my point of view, shocking that we don't give this to everybody. So squamous cell carcinoma of the head and neck has largely been a surgical disease, and I'm trained as a head and neck surgeon, so I took care of a lot of these patients. And it's mostly because there have been very few drugs that have been proven to be safe and effective for this malignancy. And I will point out that the drug that is still most widely used, which is platinum, whether it be cisplatin or carboplatin, was approved way back in 1978. It's almost always given in combination with radiation therapy. It's a radiosensitizing agent, and it has quite well-documented toxicities, which we don't need to belabor here today. 2006 was a banner year. We saw the FDA approval of docetaxel, which is not tolerated in combination with radiation and still used quite sparingly. And cetuximab, which is the first and only molecular targeting agent that's been approved for head and neck cancer. There was great hope in 2006 because of the ubiquitous overexpression of EGF receptor in human head and neck cancers, that this drug would provide a very effective alternative to toxic platinum chemotherapy. That turns out not to be the case. We completely lack predictive biomarkers, the level of EGFR expression in the patient's tumor, the level of phosphorylation of the receptor, amplification of the gene. None of these have proven to be adequate predictive biomarkers. And so today, cetuximab is largely given in combination with radiation in patients whose comorbid conditions mean they cannot tolerate cisplatin. But we, like everybody else in oncology, are now part of the immuno-oncology tsunami. And in 2016, we saw the approval of pembrolizumab and nivolumab, both immune checkpoint inhibitors blocking PD-1 for recurrent metastatic head and neck cancer. Pembro was approved in combination with chemoradiation in 2019 for locally advanced newly diagnosed cancer. And I would say that probably 90% of the clinical trials now are based on a backbone of immune checkpoint inhibitors. And so trying to figure out how a novel therapeutic strategy factors into the clinical armamentarium must take these drugs into account, which means for our preclinical models, we have to start working with immunocompetent models of squamous cell carcinoma to be able to test how these agents fit into future advances. So why should we all study head and neck cancer? And I don't think I'm speaking to an audience that needs to be converted, but unlike cancers like pancreas cancer or even lung cancer, colon cancer, head and neck cancer is very accessible for biopsy. We can do this under local anesthesia easily in the office. We have a fairly large number of well-characterized cell lines. They've been characterized by the Cancer Cell Line Encyclopedia, among other international and national efforts. It's not hard to develop patient-derived xenografts. We have nearly 100 in our lab. Happy to share them. Happy to I'll describe some data that we've generated using them. It's just important to remember that these contain murine stroma. We're finishing up a paper right now. I don't think I'll talk about it today, but we very carefully filtered out the mouse reads from our PDXs in order to mitigate the contamination of mouse stroma in the RNA-seq profile. And we found very, very little impact of doing that. So I think it's a liability. You certainly can't do immunotherapy studies because these models have to be grown in immunodeficient animals. But I think that their biology are largely reflective of the human tumor. We talked about HPV. You talked about how common oral cancer is in Southeast Asia. It's the most common cancer in India. Chemo prevention is feasible. When we see a patient with head and neck cancer, 50% of the time we'll cure them of their first tumor, but they will develop a second primary tumor at the rate of 3 to 6% a year, which means they are likely to die of another squamous cell carcinoma, whether it be of the head and neck or the esophagus or the lung. And the lack of predictive biomarkers is a theme, with the exception of PD-L1 expression as a predictive biomarker for the use of pembrolizumab and probably nivolumab. We really do lack predictive biomarkers to guide therapy for squamous cell carcinomas of the head and neck. So I had the privilege of collaborating with the group at the Broad at Harvard, led by Levi Garraway, Todd Golub, and Eric Lander. And we published this paper now about 10 years ago. And this was one of the papers that really highlighted the frequency of these notch mutations in human squamous cell carcinoma. And I look forward to the rest of the talks because it's really stunning to me that here we are 10 years later, and we still don't know how to leverage this biological finding for therapeutic purposes. Notch inhibitors haven't made sense to test in these patients, and we still don't know what to make of the presence or absence of notch alterations with respect to treating patients. We decided to focus our energies, and you alluded to this, and I'm going to talk about this for the next few slides, on the most frequently mutated oncogene, which is the PIK3CA oncogene. I do want to point out that the mutations in HRAS are now coming to the fore as potentially really leverageable for therapeutic purposes. Many of you are familiar with the farnesyltransferase inhibitor story, particularly tipifarnib. These drugs were studied long ago. They were put on the shelf for lack of efficacy, like all of our prior studies. They were tested in unselected patient populations, but we now have some really tantalizing and compelling evidence that patients whose tumors have an HRAS mutation might be exquisitely sensitive to these drugs, and there's a phase 2 trial that hopefully will be completed soon in head and neck cancer to answer that particular question. So after we published this paper, we went back, we were doing the TCGA. I had the privilege of co-chairing the head and neck group, and we had amassed a large collection, and we noticed that the genes in this pathway, in addition to the oncogene PIK3CA, were by far the most commonly mutated genes in head and neck cancer that were potentially therapeutically leverageable. I'm not, P53, of course, is the 800-pound gorilla. I can't tell you how many P53 replacement gene therapy trials I've participated on, injecting P53 genes into human head and neck cancer for not very much benefit. So we decided to dive into this story a little bit deeper. And now with the publication of the TCGA, we have now a fairly large subset. So if we look at HPV negative squamous cell cancers in the head and neck is not associated with the virus, 1/3 of these tumors will have a genetic alteration that activates PI3 kinase signaling. The majority are PIK3CA mutations. PTEN loss is also a common feature, which we studied as well. And if we look at HPV positive head and neck cancers, over half of those individuals will have a genetic alteration of the PIK3CA oncogene, making this now a fairly common genetic alteration and really amenable for precision medicine studies. So now back to the PDXs. Why am I a PDX enthusiast? Because it's so much easier to take a cell line and inoculate subcutaneously into mice. Well, here's what we found. Typically, as many of you know who do xenograft studies, the different models don't usually give us very different answers. Most cell lines are either sensitive or not to the drugs that we test in those models. But here we have a patient's tumor who has a canonical PIK3CA mutation, and we grew up this tumor, we made it into a xenograft, we implanted it into mice. And in this particular experiment, we treated it with an earlier drug, a combination mTOR PI3 kinase inhibitor. This went through clinical trials. It was stopped because of toxicity. But indeed, we find that we cured the mice with this drug. The tumors really disappeared. In contrast, when we treated mice bearing PDXs developed from stone cold wild type PIK3CA xenografts, they were insensitive. So it really convinced us that having a genetically defined model in the form of a PDX can actually illuminate the capacity of precision medicine. We then went back and we looked at the whole spectrum of PIK3CA mutations in human head and neck cancer. It looks very much like breast cancer and very different from other cancers. So for example, in colon cancer, all the mutations are canonical mutations. So we see that the three common canonical mutations in the helical domain and in the kinase domain occur in 2/3 of the cases. So it's still the most common PIK3CA genetic alteration in human head and neck cancer. But the story I'm now going to tell you that is unpublished and under review is a story about the non-canonical mutations. This is not a small subset. It's over 1/3 of our patients have these rare mutations. And as all of you who participate in molecular tumor boards, when we get a targeted sequencing or whole exome sequencing on our patient's tumor, and we find that they have one of these mutations, nobody quite knows what to do with it. And as we saw from the FDA approval of the drug alpelisib in breast cancer, it's only approved in the case of canonical mutations or recurrent non-canonicals that have been proven to be activating. So we thought it was really worth characterizing these mutations in the context of head and neck cancer in anticipation of determining how to employ drugs that selectively target nodes in this pathway. So we've developed a model in our laboratory. In this model, it's a HPV negative squamous cell carcinoma model of the head and neck, and the parent line grows under low serum conditions. But we identified a clone and cultivated a clone of this model called SD1. And when we take away serum from the media of these cells, they begin to die. So in a very simplistic way, we can introduce a candidate driving oncogene into these cells. We can take serum away, and if the cells survive, we know that that gene is conferring a gain of function. Whether it's doing so directly or indirectly is a matter of a further debate, but it meets the criteria of a gain of function alteration. And so this is what our system looks like. We use a TET-inducible lentiviral vector that we flag tag to make sure that the transfected gene is into these cells. We turn on gene production, we serum starve them, and we test cell viability. And we come up with our answers. So I'm going to get right to the punchline. So here are all of the non-canonical PIK3CA mutations found in human head and neck cancer. This was a tour de force of a very talented postdoc in the lab, Nanxing. And what she found is that about 70% of these mutations are activating. And this is very similar to what's been found in other cancers where that's been studied. So it suggests that many of these mutations are actually worth paying attention to in our patients in terms of guiding therapy. And another thing that's really worth pointing out is it appears to be cell lineage independent. So my collaborator, Gordon Mills, who's now at Oregon Health Sciences Center, has a BAF3 model, a factor IL-3 dependent model, and he characterized all these same mutations with us and independently of us. There's also the breast model, MCSF10A. Again, these are factor dependent models. And you can see that a mutation that is activating in head and neck is generally activating in these other models, and in collaboration with some scientists in Israel, they have a HeLa platform, and HeLa is also a squamous cell carcinoma model, but in this case, it's HPV positive and it's cervical. And so there's a really large concordance across different cell lineages, which is, I think, very enticing and allows us to draw some conclusions. So now, what is the clinical significance of this? And finding this patient was actually extraordinarily hard, and I'll tell you why. We typically don't get mutational data on head and neck cancer patients, and there are very few studies that allow a patient to receive a P110 alpha PIK3CA inhibitor. But we had a postdoc in our lab from Korea. He knew about this ongoing basket trial in Korea of head and neck cancer, and we found such a patient. So here's an individual with a PIK3CA mutation. Nobody knew what this mutation meant. This mutation has never been reported in any human cancer, but he had a mutation, and by criteria of this trial, because this mutation was present in his tumor, he was eligible to receive the drug alpelisib. So he failed 3 cycles of chemoradiation therapy. He developed recurrence and metastasis. He failed 2 cycles of 5-FU platinum. And then he was put on alpelisib just because he had this PIK3CA mutation that nobody knew what it meant. And he managed to maintain, he responded, and he had a durable response for a year, and then he progressed, and he's now on durvalumab, an immune checkpoint inhibitor, and he's now 2 years later, and he's still alive. So this really shows you both the CT scan of his anterior mouth and lung metastasis, both of which reduced in size considerably on this P110 alpha inhibitor therapy, which is very encouraging. So here's a patient who's telling us that this mutation matters, and he got benefit from a fairly safe and effective drug for over a year. So we now started really focusing on this particular mutation. So back to our model, we induced wild type amplification, a canonical helical domain mutation, and this novel mutation in this patient's tumor. And indeed, each of these mutations induces phosphorylation of AKT. You can see it graphically here on densitometry, suggesting that this mutation turns on the PI3 kinase pathway. Also in our screening assay, it induced growth. So here's our control, and both the canonical mutation and this and now non-canonical mutation drive growth. So it does meet our criteria as a driving oncogene. We also are looking at other phenotypes in addition to survival in our serum-dependent assay. Here is an assay looking at colony formation, and indeed this novel mutation enhances colony formation. This novel mutation also enhances cell migration, very similar to a known canonical driving mutation. We did this with all of our mutation, and here and essentially found a very high degree of concordance between survival under low serum conditions, migration, and colony formation. But now we go back to our PDX collection. So we don't have a PDX that harbors the non-canonical mutation found in the patient, but we do have a PDX that has a non-canonical mutation that is shown to be a driver across all platforms. And indeed, when we implanted this PDX into mice and treated them with alpelisib, we saw very nice anti-tumor effects. In contrast, when we took a wild type PIK3CA PDX and we treated the animals with alpelisib, we saw very little impact. Interestingly, there is a pharmacodynamic impact of alpelisib in both settings, stronger in the activating mutation than in the wild type. But this really kind of closes the loop and suggests that from this day going forward, when our patients' tumors harbor mutations that have been shown to be activating, regardless of whether canonical or non-canonical, they should be candidates for treatment with drugs that target nodes of this pathway, including a P110 alpha inhibitor. And because alpelisib is now FDA approved in breast cancer, it's easier to get it for treatment in other cancers, at least in the United States. And this is just showing some colony formation with some other mutations, the patient mutation, this mutation of the PDX, and another mutation showing that we can drive colony formation that is blocked by alpelisib therapy. In contrast, these mutations that are all shown to be non-activating don't appear to form a lot of colonies, and there's no effect on colony formation by treatment with this drug. So it's a really nice and neat precision medicine story. This is complicated, but it's really where the biology is focused. So I'm not a structural biologist, but Natalia Yura is, and she's in the building right next to me. And we engaged in this really exciting collaboration where we were able to map the activating non-kinase domain mutations onto the known structures of PIK3CA, the non-activating non-kinase domain mutations, and the activating and non-activating kinase domain mutations. There's a lot of data here. I'm happy to share it with you as soon as this paper is published, but what I can tell you is that the structural mapping provides a really nice biological clue as to why some of these non-canonical mutations activate the pathway and others don't. In addition to looking at the structure-function relationships in our preclinical models and the structural biology, we are also doing protein-protein mapping. And this is a collaboration with Nevin Krogan and Daniel Swaney from his lab, and it's been a really enjoyable and productive collaboration. And then the basic idea is if we can map which proteins are associating with which other proteins in a particular cancer context, we might be able to design drugs that target an interacting protein when the protein that we're terribly interested in either doesn't have a drug that targets it or the available options are too toxic. So we did this with all of the PIK3CA protein mutations, and we came up with a PIK3CA interactome, if you will. And I'm going to tell you a really tantalizing story. This paper is in its second revision for a really high impact journal, so hopefully the story will be out soon. But what we found was really quite exciting. Specifically, we saw that the canonical PIK3CA mutations in the helical domain had a very strong interaction with several genes, IRS1 and HER3 in this case. We focused on HER3 because there are drugs that block HER3. We did co-immunoprecipitation and indeed found that these particular mutations do co-immunoprecipitate with HER3. And in contrast, and importantly, the other common canonical mutation, H1047R, which is in the kinase domain, does not strongly interact either by the interactome or by co-immunoprecipitation studies. So this would suggest that if a patient's tumor had one of these two mutations, it would respond to a HER3 inhibitor. In contrast, if it had H1047R, it would probably not respond to a HER3 inhibitor. And so we did that experiment. So in collaboration with Silvio Gutkind at UC San Diego, we're on this grant together. We engineered these cells and did this experiment. And I will call you, so we treated these drugs with a HER3 inhibitor that has been developed by Yossi Schlesinger and now licensed to Celldex. But this particular drug, so it's a Celldex drug, was highly effective in this head and neck cancer model engineered to express helical domain mutations. The tumors were very effectively growth inhibited. In contrast, tumors grown in mice engineered to express the kinase domain mutations were really insensitive to the drug, thus very neatly proving the hypothesis that this interaction between selected PIK3CA mutations and HER3 could reveal another strategy to target this gain-of-function oncogene. So in the beginning, when we first learned about this, we were really quick to design a therapeutic clinical trial containing this particular drug from Novartis, the P110 alpha inhibitor, BYL719, or now known as alpelisib. And then we learned that it was much more complicated. So we scaled back, and now in collaboration with my former colleague, but my collaborator, Julie Bauman at the University of Arizona, we are doing a window of opportunity trial. This is a platform that I love, helped to pioneer it. We've now put hundreds of patients on this kind of a platform. The reason I love this platform, it not only allows the surgeons to really meaningfully participate in research and thus really build robust translational research teams, but it allows us to get really important information about what a drug is doing in a particular patient in their tumor. So a patient is going to have resection. In this case, we've designed the study for oral HPV positive tumors because we know over half of them will have a PIK3CA alteration. They undergo a standard biopsy and we measure their tumor by CT scan. They then get 2 to 3 weeks of daily alpelisib, which is very well tolerated, and they have their surgery, in this case by transoral robotic resection. The paired CT scans provide a proxy of clinical activity, but we really can look at the impact of the drug on the immune system, on some of our candidate predictive biomarkers, and on HPV itself. We have about 10 patients enrolled on this study. We see responders, we see non-responders. We're in the process of immunoprofiling. We are making PDXs from these patients' tumors, and hopefully in the next year or so, we'll have a lot more to tell you about the impact of this drug in the setting of PIK3CA alteration or lack of PIK3CA alteration in human head and neck cancer. So the first part of my talk was a sort of a straightforward talk. I told you about a mutated oncogene. I talked about a drug that's FDA approved now that targets that oncogene itself and how we might use it. The next part of the talk for me is more exciting, but also more complicated because it essentially leverages our understanding about receptor crosstalk and receptor signaling and how a mutated oncogene could have implications for other drugs to be brought into the clinical armamentarium, ideally in the setting of prevention, that would really make an impact for our patients. So you may be familiar with this 2012 paper from the New England Journal. It's based on the Nurses' Health Study, and this is straight epidemiology. They looked at this very large cohort of patients. They knew who had colon cancer. They knew what their survival was. They knew if they used aspirin, which is a fairly well-accepted chemoprevention for colon cancer, and they had the tumor genetic information. And it was surprising that they found that patients whose colon cancers had a PIK3CA mutation, and remember I told you these are all canonical mutations, they had a significantly improved survival if they used aspirin. So we thought, based on our receptor signaling work over the years, that we knew a lot about this, and potentially there was a mechanism to leverage this information to help patients with squamous cell carcinomas, in this case of the head and neck. So we heard earlier about ADAM17, G protein couple receptors can transactivate tyrosine kinase receptors, including EGF receptor. Part of that transactivation pathway critically flows through a PIK3CA. We also know that PI3 kinase is activated downstream of even just wild type receptor tyrosine kinase signaling. We'd studied crosstalk. We knew about the prostaglandin E2 receptor, and we knew that NSAIDs had been looked at in human head and neck cancer, but without a deep understanding of biology, and usually in combination with chemotherapy. And the results were not very informative or tantalizing. So we decided we would just test the hypothesis once and for all. And the beauty of building a center the way you are is you're going to have high quality tissue. You're going to have phenotypic information on the patients who provide the tissue. And if you also collect the information about what medications they take, either prescribed or non-prescribed, and the reasons that they do this, you will have the ability to ask this question, which very few people do, which is why our study kind of came out of the blue, because there was no other cohort that had all this information. So we had a large tissue collection. We knew whether or not the patients were on chronic NSAIDs. We were able to sequence PIK3CA. We also did FISH to look at gene amplification, which we also think is important. We looked at HPV, and at the final case, we had 266 patients on whom we had overall information. And it was a little bit of a Hail Mary. We asked the very simple question: if you're a head and neck cancer patient and your tumor has a PIK3CA mutation, and you happen to be taking NSAIDs, and in the vast majority of cases, it was baby aspirin for cardiovascular reasons, did you have any change in your overall or disease-specific survival? So here's the answer. It was pretty stunning. When we looked at our patients' tumors who were wild type without amplification, it didn't matter if they never used or chronically used NSAIDs. Their survival at 5 years was exactly what we expect. It was about 45, 50%, and there was no difference in whether in survival. And this is, I'm showing you disease-specific survival because it's more rigorous. The same thing was true for overall survival. However, if your tumor had a mutation or amplification in PIK3CA, your survival was worse. That's not surprising. That's been shown in other cancers. This is an oncogene. However, if you had the luck to have a mutated or amplified oncogene and be on a non-steroidal anti-inflammatory drug for at least 6 months after your cancer was diagnosed, your survival at 5 years goes up to almost 80%. This was independent of HPV, it was independent of other clinical and pathologic variables, and it was really quite remarkable. So we then went to our preclinical models, which are really challenging to recapitulate what you find in patients, but we think we found some answers. We had a whole suite of PIK3CA mutant PDXs. I showed you some of that data earlier, and we treated these mice with vehicle or an NSAID. We did this with Sulindac, indomethacin, celecoxib, and aspirin. We found the same thing in every case. I'm showing you the Sulindac data. So in the case of a wild type PIK3CA PDX, it's insensitive to NSAIDs. That's not a surprise. NSAIDs should not cure cancer. However, we were able to show that if it had a mutation in PIK3CA, it grew quicker. And if you treated these animals with an NSAID, you had significant anti-tumor effects, and that was pretty remarkable. That was surprising. We then asked the question why, and we really thought it had something to do with prostaglandin E2, and we think it has something to do with the immune system, because remember, the patients are always telling us the truth, and we found in our cohort that they were disease-free until their disease recurred. And it was the mutation in their tumor that ultimately dictated this pattern of recurrence and survival. So we hypothesized that it had to do with some potent circulating factors that could interface with the immune system. Prostaglandin E2 was logical. So we then measure prostaglandin E2 levels in the circulation of mice with PIK3CA PDXs. Here are individual mice with wild type PIK3CA PDXs and individual mice with mutant PIK3CA PDXs. And we see a significant upregulation in the blood of the mice who are implanted with patient-derived xenografts, either wild type or mutant for PIK3CA. I then mentioned the immune system. We then engineered some models, and now we're growing them in immunocompetent mice. These are MOC1 cells that are very well characterized and known to respond to some degree to immune checkpoint inhibitor. And indeed, when we engineered the cells to express high levels of PIK3CA or this canonical mutation, we saw a significant decrease in NK cells in the tumor. And we saw a similar decrease in CD8 positive T cells. So we now think there's some potential immune mechanism to explore in patients that we're going to test in our patients on the ongoing window trial. But the definitive trial really needs to be an adjuvant trial where we cure patients of their head and neck cancer. We have the genetic information about their tumor, and they're randomized to receive NSAID or placebo for 2 years. We are modifying this slightly. There's some really interesting data coming out of the colon cancer world. They did a study like this in colon cancer, but they were, for various reasons, compelled to use celecoxib. They found no impact on celecoxib. We don't think that there's any evidence that it's COX-2 specific, so we would not want to use celecoxib. And we think there's a lot of good reason actually to use full dose aspirin as opposed to baby aspirin. And so we're modifying the trial design and looking for a possible way to fund this trial, because without a pharmaceutical supporter, it becomes a really challenging economic enterprise. So I know it's tempting to say, oh, you've already shown it in a retrospective cohort. Can't you just take a baby aspirin or an aspirin a day if your tumor has a PIK3CA mutation? Sure, but all of the really compelling data show us that aspirin is not without toxic side effects, particularly bleeding, particularly in elderly patients who have no compelling indication to take it. So I'm hopeful that someday someone will do the right prospective study and definitively prove whether the presence of a PIK3CA alteration in a squamous cell carcinoma is actually a predictive biomarker for disease prevention and prevention of recurrence and new primary tumor formation with aspirin. And that would be sort of a major benefit to people for all the right reasons. So in the last few minutes, I'm going to switch gears. PIK3CA is an oncogene. They're drugs that target it directly or indirectly, but transcription factors are much, much harder to target. So I'm going to tell you a little bit of story about a driving oncogene that is STAT3 in human squamous cell carcinoma. So STAT3 is a very attractive cancer target for 2 reasons. It's overexpressed and hyperactivated in the majority of solid and hematopoietic cancers by the tumor cells. So it has a phenotype on the tumor cells, but in this era of immuno-oncology, it is also hyperactivated in tumor infiltrating immune cells where it is immunosuppressive. So if we actually had a drug that could block STAT3 in the tumor, we could enhance anti-tumor immunity and increase tumor growth, and that would be quite a home run. So adding fuel to the fire was this paper from Jeff Settleman when he was at Genentech that didn't go in to find STAT3. He was really looking at drug resistance mechanisms in the setting of tyrosine kinase addicted cancer cells, and found STAT3 that emerged as the primary mechanism of drug resistance, which is actually how we got to it in head and neck cancer. It was in our patients who were resistant to cetuximab. It was hyperactivated, and hence we started to study it. Now, the mantra is that transcription factors are undruggable, but I hope in the next few minutes to convince you that that's not entirely true. So Most folks have tried to block downstream signaling using upstream inhibition of either receptor tyrosine kinases or non-receptor tyrosine kinases. We've looked at all of these strategies. They work, but there's a rapid emergence of drug resistance, and it's not the most effective STAT3 inhibitor. I won't show you any of that data because I want to get right to our STAT3 inhibitor story. So being a naive surgeon, I created a drug. I don't know if I had known what I know now about drug development, if I would have done this, but it seemed like the right thing to do. We'd read a paper about a decoy blocking STAT6. It was not difficult to synthesize or design the drug. We very carefully made it as small as possible, so it's considered a large, small molecule. We were able to show that it bound to STAT3 dimers in the cytoplasm. It crosses into the nucleus. It binds to the promoter regions of STAT target genes. And this kind of drug is really a first in class, and there's no really plausible mechanism for drug resistance, and we haven't seen drug resistance. So our first generation molecule that we took through to a phase 0 clinical trial looks like this. We had phosphorothioate modifications on each end. In our preclinical work, we have a mutant control version that just differs by a single base pair, but does not bind to the STAT3 response elements in the promoter genes. We and many, many other people have studied this. Its significant limitation is it has to be injected into the tumor because it's not stable in the circulation. So this is what we did. We did a clinical trial. I took my patients to the operating room. I randomized them. We biopsied them in the operating room. Half the time we injected the tumor with our STAT3 decoy. Half the time we injected the tumor with saline. And then anywhere from 2 to 6 hours later, after the neck dissections were complete or the flap was in place, we took out the tumor. And it was a very simple question, pharmacodynamic. If we inject the decoy into the tumor compared with saline, do we see evidence of downregulation of STAT3 target gene expression? And the answer was yes. So we chose, in this case, BCLXL and cyclin D1. There's just 2 of the many STAT3 target genes. And you can see that in these 4 patients who got the decoy, there was lower expression following decoy therapy with a very brief exposure. We did immunohistochemistry on the large cohort, and this was 15 patients got decoy, 15 got saline, and we found this significant difference, and we published this. But I was done with an intratumoral formulation. So with the collaboration of a chemist who specializes in nucleic acid therapy, Daneth Lee at Carnegie Mellon University in Pittsburgh, we went through many iterations, but we ended up with our decoy stabilized by the addition of these carbon spacers on the ends. And now this is really a drug that can be given intravenously. So we were able to show that there's really a nice dose response in a xenograft model. And we've experimented with the frequency of injection. What I can tell you very confidently is you get the same anti-tumor efficacy with twice-a-week injection as you do with daily injection, and we're now exploring once-a-week injection. And we find no benefit to going above 5 mg per kg of the decoys. And these are all tail vein injections in these mice. A reviewer asked us to prove that it was better than an FDA-approved drug that blocks STAT3, in this case, ruxolitinib. And indeed, here's ruxolitinib, and here's our decoy down below, so it is. And they asked us, how does it compare to cetuximab? Cetuximab's pretty good in the xenograft model. Our decoy was a little better, and indeed, when we combined the two, the tumors disappeared. So that was encouraging in terms of future translation. But now, of course, it's the era of immune checkpoint inhibitors. So this is our first experiment using an immunocompetent squamous cell carcinoma model. These were the MOC1 cells. We combined our decoy with the murine version of anti-PD-1. And indeed, we see that we can get a significant improvement in percent Treg downregulation when we give the decoy in combination with the checkpoint inhibitors compared to our mutant control decoy and the checkpoint inhibitors. So we're continuing these studies and looking at the immunoprofile of this approach. In addition to downregulating those aspects of the immune system, we increase interferon gamma expression with the combination of the decoy and anti-PD-1. So we're hopeful that's the way to go. But I want to finish by telling you that it's not just squamous cell of the head and neck. Here's a study in a squamous cell of the lung by my colleague Jill Siegfried. This has been published. She saw anti-tumor efficacy of the cyclic STAT3 decoy in her models of EGFR mutant tyrosine kinase inhibitor resistant non-small cell lung cancer. But maybe the most tantalizing data is the following. And this came from her graduate student who decided when he was doing a carcinogenesis experiment with NNK just to see if the decoy had any impact on tumor outgrowth. But he didn't remember that he'd treated the mice with the decoy, and these take months and months for the tumors to outgrow. And one day Jill gets a call that she's got a cage of mice that can't breathe and a cage of mice that are perfectly fine. Can she come take care of them? And the mice that were perfectly fine were the cyclic STAT3 decoy mice, and the mice that had lungs filled with lung cancer were the cyclic STAT3 mutant mice. So they'd gotten the inert control. And so we decided to expand on this work. It's now been published, but we are taking this idea of prevention. The NCI is now funded through its Prevent Network, creating an aerosolized version of the STAT3 decoy. So Jill is now working on that. And the idea is that you could inhale this to prevent conditions that are dependent on STAT3, including lung cancer, and now really interestingly, probably COVID-19. So lots of applications with the inhalation format, just early days of starting that. But the final piece I want to close on is the opportunity to do clinical trials in patients who aren't people. And my sister is a veterinarian, and she has regaled me for years with the tragedy of cat oral cancer. So pet cats get oral squamous cell carcinomas, and they all die. They get radiation. The radiation isn't effective. Their oral cavities are too small to tolerate the excision. And so we looked at, in collaboration with veterinary pathologists at the University of California, Davis, which is just about an hour away from San Francisco. And indeed, STAT3 was hyperactivated in about half of those tumors. And when they look at the tumor microenvironment of their cats with oral cancer, it looks very, very much like human head and neck squamous cell carcinoma. So we now are about to open a clinical trial in pet cats. We were able to fund this with an R01 because unlike a clinical trial in people, you just need IACUC approval. You don't need an IND. So the manufacturer is ongoing and we hope to open this at UC Davis in the next few months, but it's going to be a phase 1-2 trial of the cyclic STAT3 decoy, 2 pet cats. And even though reviewers keep forgetting that these are not laboratory animals, I keep reminding people that we can't do things to these animals like we do in our laboratories because these are members of people's families. But we are going to access the tumor and the tumor microenvironment and hopefully get a lot of information that could help us when we're ready to roll out this treatment for patients, human patients with squamous cell carcinomas. So I'd like to conclude by thanking you for inviting me. I only wish I could be there. Please invite me again when you're open for business. I'd love to come. But I think in cancer research, we really are at a confluence of a deep understanding of biology and genomics, and we really have to grab the bull by the horn, identify predictive biomarkers, even if it's hard. So there great opportunities. The challenges are immunotherapy is here to stay. We have to contend with that in our models and in our new treatments. And I just want to emphasize the incredible importance of tissue biopsies on treatment, particularly in the setting of resistance to identify underlying mechanisms. And I want to end by thanking all of my collaborators and funding sources at UCSF and elsewhere. And thank you very much.